Cell Reproduction, Chapter 9. Body Heal Thyself. Bartolo Colon, he was a pitcher. He had a great fastball, but eventually he injured his shoulder and he couldn't really continue pitching. And so, of course, as we all wonder, well, why is it that the body can't just heal itself in every situation? For example, you get a cut and the body can heal itself. You break a bone even and the body can heal itself. But in this case, his body couldn't really heal itself, apparently. So you got to ask yourself, how does the body heal itself when it is successful healing itself? And ultimately, it is by cell reproduction. The destroyed cells that have died, are replaced by other cells. But some parts of the body, they just have a harder time replacing lost cells. So in 2005, ligaments and tendons were damaged. So ligaments and tendons, those are extracellular protein structures, so outside of cells, and they're made of protein. Basically, collagen is a kind of protein. And so it's not cellular in nature. So your muscles, your brain, those are made of cells. But ligaments and tendons, those are basically things that cells have made. So cells have made them. They can repair them to some extent, but not if the injury is significant enough. So in 2010, what they did was they injected some stem cells from bone and fat from him. So they took cells from other parts of his body and they injected them into the area of injury. 2011, well, he was signed by the Yankees. 2018, as recently as that, he was still going strong. So the idea was these stem cells that were injected in 2010, that they would somehow bolster the cellular repair mechanisms. And we'll learn a little bit more about how that may have happened. Remember, we don't actually know that the stem cells cured him in this case. This is a fairly experimental therapy. And so we're not really sure. We know he got better, but the thing with disease is quite often we just get better if we rest for long enough. So we're not really sure if the stem cells work. So what are the functions of cell division? The topic of this chapter. Cells reproduce by division that produces two identical daughter cells. Two identical daughter cells. Why are they identical? Because DNA replication is close to perfect, except mutations. When we covered that in a past chapter. Identical because each daughter cell gets a complete copy of every chromosome and also every gene. So that's what we mean by identical, genetically identical. So the cells might be slightly smaller right after division. They may have a somewhat different number of mitochondria or chloroplasts, but they are still genetically identical. Why do cells divide? Number one, organismal growth. So when a plant is getting bigger, cells have to divide so that the plant size can increase. Development is another one. Organismal development. Taking a look at the first single-celled stage of a human being, and it's just a cell. It's not like it's a tiny cell that has an arm and a leg. It divides, 2 cells, 4 cells, 8 cells, 16. Still no real development. This is still really just growth. But eventually, a little after this blastocyst stage, and even here, but it's kind of hard to tell unless you're like this is your specialty. Cells start to take on specific functions. So by 4 weeks, there's a lot of development that has happened. There will be some cells that are involved in bone, others that are involved in muscle, nervous tissue, the works, and then development continues through 10 weeks and on throughout life, more or less. Reproduction. So some species will reproduce asexually. That means that there's only one parent, and we'll talk a little more about it later. Shown here, this cell is going to reproduce itself by dividing, and I don't really like this diagram that much. It's showing that just one cell's been produced. If it divided, though, we would have 2 cells. So there'd be another one here with those X's are representing chromosomes in there. And sexual reproduction is also a kind of cellular division, which we'll talk about in a later chapter. These 2 cells represent different parents. So if we can use the easy example, mom, dad, human beings are all familiar with what these words mean. Mom and dad both produce a sex cell, each one with half the amount of genetic information as the parent cell in the testes or the ovaries. And then these two cells will fuse, that's called fertilization, and we get a single offspring, well, usually a single offspring. And the offspring is not genetically identical to either mom or dad. It's a mixture of both of them, 50% mom, 50% dad. Next, to repair damage. So you injure yourself, the way that your body is going to heal itself mostly is by producing new cells to replace the ones that were destroyed and to heal the tissue that was damaged. So this slide could really represent repair of damage or maintaining tissues. After all, the skin is a tissue, so we could be repairing it. And last one here, maintaining tissues. Looking at the digestive tract, this is a good example of maintaining tissues because the interior of the digestive tract, especially the stomach, for example, cells die really rapidly and have to be replaced. So the stomach is a very acidic environment, so cells are lost really quickly. And the intestine cells are lost quite quickly as well. And so they need to be replaced. If they're lost, they must be replaced. So that's maintaining tissues. And on this topic, I wish we could do a whole course about just maintaining tissues, because what that's about growing old. And scientists are beginning to do some research to make some breakthroughs, rather, where they may be able to either slow aging or in some cases even reverse aging. Depends on what scientists ask. Some of them are very optimistic and some of them are thinking, probably not going to happen. Stem cells and differentiation. So these are the cells that continue dividing throughout our lives to replace lost cells, do all the things that cell division that we've talked about that cell division is for. There's a couple of kinds or classifications of cells here that we should go over though. So self-renewal. Some cells retain the ability, stem cells, that retain the ability to continue dividing forever. So looking down here at this diagram, this is a stem cell and it can continue dividing. Other cells, such as a non-stem cell, so this up here, this stem cell divided once, and one of the cells is going to basically stay a stem cell, not be differentiated. It'll be very general, just kind of a circular little structure, and it doesn't actually do anything. The other cell here, so the non-stem cell, will retain a limited capacity to divide, and it could divide to form a couple of different types of cells. This is a little bit of an exaggeration that one stem cell could divide to produce nerve cells, muscle cells, bone cells, and blood cells. Usually there are just kind of classes of stem cells generally. So there might be one class of stem cell that can replenish all of the blood cells, the cells that are in the blood, red blood cells, white blood cells, etc. Then there might be another class of stem cells that can reproduce to replenish lost muscle cells, etc. But the details of that you do not need to know. Okay, so permanently differentiated cell, that's right here. So those are cells that cannot divide, and some good examples would be blood cells, red blood cells specifically. They don't even have a nucleus, so they could, if they were somehow able to divide, their daughter cells, I mean, they don't have any genes or DNA, so the cells would have no instructions to how to do anything. So they cannot divide, very obviously. These nerve cells as well, they cannot divide. So a nerve cell, some of them, they go all the way from your toe to the spine, to the base of your spine. And so they don't divide. They are simply there. To some extent, they can be replaced, and that is still under debate as to how many of them can be replaced in what circumstances. Very recently, if you looked at a textbook, and I think our textbook says this as well, so you're free to say that. I'm not going to be trying to trick you on an exam or anything. They're saying brain cells cannot continue dividing in any way. But we are finding that, in fact, there are some stem cells lurking in the brain that will continue to replenish the brain's supply of cells, but it's a little bit more limited. Same thing with the heart. Somebody has a heart attack and it's kind of a severe one. They can lose some of the cells in their heart, and generally that's going to result in a close to permanently weakened heart. However, so an example of stem cell therapy, It's possible that in the near future or perhaps distant future that we could inject a person's own stem cells onto the surface of their heart, or I'm not sure exactly how a doctor would do it, but they'd get them in there somehow, and that those stem cells would replace damaged cells of the heart. So the potential for stem cells as a therapy is tremendous. So stem cells, again, they are undifferentiated cells. Undifferentiated means that they don't have a special function. And when they're stimulated to divide, they divide by mitosis to do all the things we talked about. Replace dead cells, repair tissues, provide new cells for growth, or new one, immune system cells. So when you feel like your neck's a little bit swollen, for example, in part that is because cells, white blood cells, are replicating really, really fast, and because they're replicating so fast, they're growing, and so that area of the body gets a little swollen. Okay, so stem cell line, these fellas down there, all cells here are going to remain undifferentiated, and then the permanently differentiated cells, they're differentiated. That means they have a function. So starting out from a zygote, this zygote can obviously become any kind of cell in the body because it does become every kind of cell in the body. Can become all 200 different cell types in the body. Are scientists doing research on zygotes or embryonic stem cells? If you read about that in the news, it is a controversial issue because a zygote can become an entire human being. So what's the future of research here? Well, the world is a big place, and China, they don't have too much hesitation to do research on embryonic stem cells. And so they are doing a lot of research on embryonic stem cells. Here, I believe it was George W. Bush who put some limitations on them. So it's a controversial issue. And it's a reason why we should be scientifically literate so that we can make decisions based in part on science. So zygote, coming back to our textbook information, zygote can divide, and one of these cells will go towards down the path of differentiation. So it's going to go do something important in the body. The other one just stays a stem cell waiting for some stimulus to continue dividing. So divide again. I made this animation myself, so it's not that amazing. So another division results in some more cells that are going to go do things. So if we're talking about a whole organism developing, these differentiated cells could be anything: skin, eye, bone, you name it. And then division continues, and point being is these stem cells will stay in the body and wait until they are needed to replace lost tissues, replace lost cells or repair damaged tissues. And this is a picture of a fetus, perhaps the most iconic example of cellular differentiation and also growth. This single zygote, incredible ability to divide. The genetic programming in there tells it when it needs to produce more bone, eyes, you name it. It's pretty amazing. Sexual reproduction. Briefly going to talk about it. It's a topic of another chapter. So sexual reproduction produces offspring which are not identical to their parents, and there are always two parents. It takes two, and it can't be done with three, not in any species. Asexual reproduction produces identical offspring from a single parent, so they're going to be genetically identical. When does this happen? In bacteria, most commonly single-celled eukaryotes. For example, yeast, the yeast that makes beer, alcohol, wine, all that stuff, they can actually do both. They can individual cells will sort of fuse and exchange and mix up some DNA and then divide, but they can also just divide all by themselves to produce two offspring cells that are genetically identical. Plants, they can do both. So for example, a potato, if you have a potato, you can pick it up, you can look at it, and those eyes, they're usually called eyes, and that's not a scientific word. If you cut a piece of that potato off and it has at least one eye, you can bury it and it will grow into an entire new plant. So you could take one big potato and divide it a few times and make 5 different plants. All those plants will be genetically identical. On the other hand, that same potato plant will grow flowers, and flowers are used for sexual reproduction, pollination. So they can do both. Kind of neat. And some plants, you can take a cutting of them and put it in the ground and it'll grow. and that would also be asexual reproduction. Animals, very rarely. There are some examples in lizards where the females decided, you know what, girls, we don't need males anymore, so we're just going to get rid of them. That's actually not what happened. Evolution is what happened, and it turned out that, well, males don't have offspring themselves, and a few groups of lizards, their environment was so stable, they didn't need to produce different types of offspring for an ever changing environment to hedge their bets on who knows what kind of environment my offspring is going to face. The lizards were super adapted to their environment, and so it, via random mutation, the females develop the ability to reproduce without having sex. They just lay an egg. And it's really quite a wild story. The females engage in a sort of physical behavior akin to sex, and their hormone levels will even change, reverting from male to female based on what position they are in during the kind of like fake sexual act. And then after this fake sexual act, the lizard in the female position will lay a few eggs. Quite a wild example, I know. Asexual reproduction in plants. So aspen trees are a good example because look at this grove up here, grove of aspens. They will send basically roots underneath, underground, and then a little distance away, they come up and grow into a whole new tree. So they're all essentially one single organism. You can cut them off from one another, and then they're two organisms, but they are still genetically identical. Then down here, this would be another single tree. I mean, I guess you could say there's whatever 50 trees, but they're all genetically identical and may even be still connected to each other. These are huge, huge organisms. And then this is another tree down there. Some fungi will do this as well. I think technically the largest organism on Earth, I think it may even be in Oregon. Just kind of an interesting side note, but it's a fungus, and it lives underground, and it just spreads underneath the soil and remains interconnected via cells. A couple other examples: single-celled organisms. So right here, this is a single-celled paramecium, and it'll just divide itself in two, and that's reproduction. Bacteria, it divides itself in two, that's reproduction. And some little animals, I gave you the example of the lizard, that's the most interesting example. This is a hydra down here, and when it's ready to reproduce, it just kind of grows a baby hydra off the side of its body, so a bud. How do prokaryotic cells divide? We'll look at this in a little bit more detail. Basically, after a long growth phase, prokaryotic cells reproduce by a means called prokaryotic fission, also called binary fission. This is maybe a little easier to remember because they are prokaryotes. So looking at it in this diagram, let's take a look at the top left here. So the prokaryotic chromosome is just a single loop of DNA, and you can see it in yellow here. That's the chromosome. The process of reproducing for bacteria or prokaryotes is really simple. So basically, what they do is they copy their DNA, as shown here. So now you can see there's two chromosomes present. Eventually, the two chromosomes are spread to opposite ends of the cell. And then looking over here, I'll circle it, the cell pinches inward in the middle until it closes off, and there are two cells produced. It is more simple in part because there's just a single chromosome and it's a circle, whereas down here on the right, this is a diagram showing all of the chromosomes in a human being. Human beings have 23 pairs of chromosomes or 46 individual chromosomes. You can see number 1 here. Chromosome number 1, there's two copies of it. One comes from mom, one comes from dad. So to reproduce all of this DNA and then make sure that each daughter cell gets a copy of every one of these chromosomes, and also that each daughter cell gets only one copy, not two, is a little bit more complicated. So we'll spend more time talking about that. Eukaryotic DNA organization. There is an animation. There's a link here, I believe, in the PPT should be there. It's also on Blackboard, so you can take a look at an animation that shows how DNA is packed or compacted. Just a couple of bullets here. DNA must be condensed or coiled to fit into the nucleus for easy manipulation in cell division. The idea is essentially this. Consider when you go on vacation, you are going to fold up all your clothes, put them in a suitcase. While they are in the suitcase, you can't use them, but it is much more easy to transport them. So that is essentially what's going on with DNA when the cell divides. Looking at the top or upper portion of this diagram, DNA is a long, thin molecule. And then as you can see, as it winds down here, those yellow balls, those are histone proteins. And DNA will wind around those histone proteins. You check out the animation, it'll be a little bit more clear, hopefully. And after it winds around the histone proteins, then these units, they're called nucleosomes, they themselves then start packing themselves, and it's called supercoiling. So the DNA coils up, and then you can see it coils again, and the animation will really make this a lot more clear. All you need to know, it coils, it compacts, it's more condensed. So bottom line, last diagram down at the bottom here, a couple of fat chromosomes right here. And I'll explain why it looks like there's kind of two, sort of one on the left here and then one on the right. But anyway, it's short, fat, and that's because it has been super condensed, super coiled and condensed. Eukaryotic chromosome structure. So now let me bring those out there. Okay, so this is what a chromosome looks like, and one single chromosome will only look like this. On the left, forget that word chromatid for just a moment, and then when DNA is copied, so DNA synthesis, DNA replication, the replicated chromosome is right next to the original. So we could call this the original. And then this one is the new chromosome. And they remain attached at this place called the centromere here, and that keeps them attached. A little bit more on why that is in a moment. And what else? Right, after this, so after DNA replication has occurred, we call this X-like structure a pair of sister chromatids. So both of them together, 1, 2, is a pair of sister chromatids, and one of them could be called a chromatid. All right, so let's see. Bullet number 2 here, the centromere, which I mentioned, is a constricted region, and it holds the two sister chromatids together, and it's also where the spindle fibers attach. We'll learn a whole bunch more about spindle fibers in a bit, but basically, to give you a preview, spindle fibers are going to attach right here, like go that way, and then another one here, and go that way, and they're going to pull these two sister chromatids apart, and they'll pull them in, towards opposite ends of the cell. And in that way, we can make sure that each end of the cell receives one copy of each chromosome, because this is one copy, there's a second copy, they're identical. So if they're attached at the centromere, it's an easy way for the cell to organize copies of each gene. Telomeres. Telomeres are protective caps found at the end of chromosomes, and that's really all you need to know about them. But what does this mean, protective caps? They're DNA. They're not something else. They tend to have more G-C base pairs, and the reason for that is G and C are held together by 3 hydrogen bonds as opposed to 2. So that helps the ends sort of stick together so they don't unzip like a zipper that doesn't have a sort of cap at the end that stops that from happening. Also, the telomere does not have any genes, so if any of it does sort of get damaged or unwind or unzip or break off, it's not going to be a problem for the cell, because the cell can't start losing genes. The eukaryotic cell cycle. So the eukaryotic cell cycle we can see is a little bit more complicated. Quick overview with this diagram. There is a G1 stage, there is an S stage, and a G2 stage, and then this whole thing in blue, this is sometimes called M phase or the mitotic cell division phase. That is when the nucleus is dividing. So progression through the cell cycle in multicellular organisms is variable. It's different in different cell types, different organisms. Some cells may exit the cell cycle and never divide again. We talked a little bit about that earlier. Some brain cells, red blood cells, and some cells may enter or continue through the cell cycle and divide in response to things like growth hormones. And if you're wondering where do these growth hormones come from, one good example is, well, exercising. Sometimes some cells are stimulated to divide when you exercise. It's one of the reasons that exercising is really good for you. So, quick overview of one part of the cell cycle, which is the main part, interphase. Interphase basically means whenever the cell isn't dividing, this is what's happening. So interphase, between the division phases. So it is everything in yellow here. So all the way around here, that is all interphase. So it consists of G1, or growth phase. This is essentially when the cell is going to be doing whatever it's supposed to be doing, being a red blood cell, being a skin cell. And if it's a cell that can't continue to divide, then it would simply stay in G1 forever until it eventually dies or is destroyed by something. So G1 is growth, differentiation. It may divide if signaled to. So stem cells, those cells that are just sort of waiting around to repair damaged tissue, replace lost cells, etc., they will be in G1, essentially waiting for a stimulus to divide. So if they're signaled to divide, one of these stem cells, we'll call this a stem cell here, if it is signaled to divide, then it will enter into the next stage of interphase, and that is S phase, which stands for synthesis, specifically DNA synthesis. Pardon me. So if a cell goes from G1, here's G1 to S phase, that essentially means this cell is going to divide now. It's not going to replicate its DNA and then sit there waiting for other stimuli to cause it to continue through the cell cycle. It's now going to divide unless something goes wrong. So S phase, DNA is copied. The chromosomes are all copied. So we would move from one sort of big fat thing like this to two. That's DNA replication. Now we've got two of them connected by the centromere. And in G2, not a whole lot happens here. G2, growth phase, it's almost not like the greatest description, really. All that happens here is the proteins that are necessary for cell division will be made. A bunch of different proteins. I'm not going to go through the specifics. The eukaryotic cell cycle continued. So nuclear and cytoplasmic division. The cell has to divide two main things. The first thing that must be divided is the nucleus, specifically the DNA in the nucleus. So we've already copied all the DNA in S phase. Just as a reminder, we've copied it. So now we have two copies. During mitosis, the division of the nucleus will make sure that each of the two daughter cells gets one copy of all of the DNA, all of the chromosomes. So the nucleus divides. Briefly, there will be a cell that essentially has two nuclei. So one nucleus here and another nucleus here. And yes, I know it kind of looks like some kind of like alien, but no, two nuclei. The second thing that needs to be divided is everything else in the cell, and that's called cytokinesis. Cyto means cell, kinesis means movement. Division of the cytoplasm and organelles. Cytokinesis is pretty simple. The cell divides, like so. Then we have two new cells, each with its own nucleus, like so. More on that later. Body, heal thyself. So ligaments and tendons don't tend to heal very well because they are not really made of cells mostly. Hypothesis: if we inject stem cells into the injured shoulder, they will divide and heal the tissue. Okay, just a little review of the scientific method here. And the problem with this is as follows. Do we know that the stem cells injected into his shoulders are the causative factor that caused his shoulder to heal? We don't know because there's no experimental control. There's no experimental control. So this is really new science, experimental science. And for this to be solid science and just sort of to be generally offered to anybody who has a shoulder that's not working that well, we would need to do a proper experiment, such as we get 200 people who all have problems with their shoulders and the ligaments and tendons in their shoulders. We randomly put half of them into one group. We give them some kind of a placebo, so we give them an injection of salt water, and then the other half, we give them an injection of stem cells, and then we see if there's a difference. That is how all medicines must be tested, because here's the thing. I mean, consider the common cold. We all have various things that we feel make us feel better or maybe even heal us. The thing is if you just do nothing when you have a common cold, you will get better eventually. So the same thing may be true with many injuries. If you do nothing, your shoulder may simply get better on its own. That is a fact. So this was not a scientific experiment. It is simply anecdotal. He got this treatment. He got better. It is still of interest. Just because it wasn't proper science doesn't mean it is worthless. Just like many other alternative medicines. Just because there's no experiment doesn't mean it doesn't have value. So if it seems like it works, then what scientists would, should, can do is do a proper experiment with a control group. Okay, so class question here. When they divide, how will each daughter cell get a complete copy of every chromosome? That is what we're going to talk about next. And I need to rewind and talk about this diagram over here just very briefly. So these are the tendons. They connect muscles to bone. And ligaments, those connect bone to bone. So those would be these fellows right here. And these tissues, just notice they're not red. And that's true. They're not red, even if you opened up a shoulder and looked at them. And that's because not a lot of blood gets to them. And that is why they don't tend to heal very well. They don't have a good supply of blood. So getting the nutrients necessary to repair them is difficult. And there's not a lot of living cells in there. It's mostly just proteins, fibrous proteins connecting one thing to another thing. Muscles, on the other hand, they have a really good blood supply, and so they will typically heal much faster than a ligament. The eukaryotic chromosomes. So, again, a eukaryotic chromosome, this is what it would look like right here before DNA replication. There's these telomeres. We're not going to talk too much about them now. And then there's the centromere. So the centromere is where, after replication, the centromere will hold the sister chromatids together. So look at the second diagram down here. And we've now copied the DNA. This would have happened in S phase, which is part of interphase. It happens in S phase. And these are called sister chromatids. They are 100% identical, totally identical. And each daughter cell after division needs one of these two copies. So after division occurs, we're going to see what we do see here. You can see they're not connected to each other anymore, and each daughter cell will get one of them. So this one will go that way, and then the one on the bottom will go the other way to opposite ends of the pole, and then cytokinesis occurs, dividing them into two different cells. Events of interphase continued. So cells prepare for mitotic division. During interphase, chromosomes are replicated in S phase. What I said on the last slide, down here. We go from one chromosome to two sister chromatids, which is just the result of DNA replication, which we already talked about in another chapter. Necessary proteins are synthesized in G1 and G2. Mostly we talk about G2, these proteins getting synthesized, but G1 as well. And in interphase, if you look at a picture of a cell in interphase, it can be a little bit difficult to determine if the cell is in interphase. The biggest clue is that there will be a nucleolus present during interphase, and that is a denser part of the nucleus shown right, let me draw an arrow to it, right there. So that would be the nucleolus, and that is present in interphase and disappears in prophase. If you asked me to tell you what phase this cell is in, I would be able to tell you that it is almost certainly in interphase, but I can't see a nucleolus there, and sometimes it's just not visible. It may be hidden behind something, or the focus of the microscope isn't focused on the nucleolus. It's a three-dimensional structure, a cell is. So it's not always visible. Generally, it is, though. Events of mitotic prophase. This is the first phase of mitosis. Let me also introduce all the phases just so you kind of get a preview. There's going to be prophase. I'm not going to write them all out. Prophase, that's the one we're going to talk about here. Then metaphase, then anaphase, and then telophase. So there's just 4 phases. This is the first one, and it is also the longest one. Okay, so in prophase, duplicated chromosomes condense. They begin to be visible as well as individual chromosomes. The nucleolus disappears, so the denser region inside the nucleus disappears. Spindle microtubules form. And on the topic of spindle microtubules, these microtubules will attach to the centromere. So let me draw a little on this diagram here. The spindle microtubules and the spindle fibers, those are the same thing, just two words, two different words. So they're shown in red here, and they originate from opposite poles of the cell. If you can see me kind of clumsily drawing here, they originate from opposite poles of the cell. So the bottom pole right here on the bottom, it's going to send out all these microtubules or spindle fibers, and it's sending them out blindly. You can imagine maybe a blind Spider-Man just shooting out webs in random directions. That is essentially what happens. Not shown here. They'll send them out in totally the wrong direction, like that direction as well. Hit the cell membrane, and then they just kind of fall apart. Erase that. Okay, so there's the opposite poles of the cell. They are sending out these microtubules, and they're sending them out until they hit the kinetochores on each side of the pairs of sister chromatids. So if I drew the microtubules here, this would be one, and it's going to bind to one side of the pair of sister chromatids. Would be another one from the other pole of the cell, pulling in the opposite direction. So once they have bound, they can pull and push until they get all of the chromosomes or pairs of sister chromatids negotiated into the middle of the cell. That will be the start of the next phase once they're all in the middle of the cell. The spindle, a little more detail on what I was just talking about. The mitotic spindle is microtubules. Polar fibers extend from one pole of the cell to the opposite pole. So they start on one end of the cell, also the other end of the cell, and they're just kind of blindly shooting out from opposite ends until they bind to the kinetochore on opposite sides of the sister chromatids. And that is shown here with a little bit more clarity than the last slide. So spindle fibers, they bind to the kinetochore, and that's going to allow them to eventually negotiate all of the pairs of sister chromatids to the middle of the cell. A little bit on identifying what phase a cell is in. You look at this and you're going to think, wow, that's really hard to tell what phase these cells are in. This is a different phase we haven't gotten into, so I'm not going to talk about those. But let me use one of these cells. That's right here, the one I've circled. This cell is definitely in prophase, and the reason for that is this. It looks very chaotic, except for one thing. You can see individual blobs more clearly, and that means that the DNA has condensed, and that's one of the main features of prophase. Compare this cell. I'll call this cell number 1, just so we've got a reference. Number 1, compare that one to this cell down here. It's the DNA, the chromosomes are not that clear. It's hard to identify a single kind of chunk. Let's compare it to this cell. Cell number 1 compared to this one, it's much more clear seeing the individual clumps of DNA. And all the rest of them, except for the ones I scribbled out, they're also much less clear. This one right here could give me a run for my money identifying whether or not it's in prophase or interphase. Don't worry, I will not be that mean to you at all. And also remember that these cells are not 100%, these stages are not 100% distinct. For example, it's when does a child become an adult? It's not like it happens at one point and then you say, aha, that's clearly an adult and not a child. Same thing with these phases. So when we look at real pictures like this, that's why it's a little vague sometimes. So I won't at all be tricking you. All the ones I show you will be very clearly in one phase or another. Events of mitotic metaphase. So this is the next phase, meta, middle. So all the chromosomes are in the middle of the cell, and each sister chromatid is kind of oriented to facing an opposite end of the cell. Spindle microtubules have attached to the chromosomes' kinetochores. They lengthen and shorten, like I said, to negotiate them into the middle of the cell. chromosomes pulled into a straight line at the equator of the cell, and each kinetochore of every duplicated chromosome faces one side of the cell. So shown here, one of the sister chromatids that has its kinetochore bound to these microtubules um, is facing upward. And then the other sister chromatid with its kinetochore is facing downward, so it can be pulled down. You can see here, this is a real picture of metaphase, and this I would call pretty clearly metaphase. Yes, I could ask you to identify what phase is this in, and it'll be easier once we actually see all the phases, right? But this is not chaotic. It's not anything like the cell I showed you in the last slide, where prophase is kind of showing these blobs. Clearly, the chromosomes are in the middle of the cell or in a line. Metaphase. Chromosomes, two sister chromatids, line up at the equator of the cell. At this point, I'd recommend checking out this animation, and there's a link to it on Blackboard for an overview of all the phases of mitosis. I've introduced prophase and metaphase, and I would say you should preview it now. So take a look at that, and then we'll continue. Again, metaphase, all the chromosomes line up in the middle. Eventually what's going to happen is they're going to get pulled to opposite ends of the cell. If you watch the animation already, you'll have seen that that has happened. And that is called anaphase. Microtubules pull the sister chromatids to opposite poles of the cell. There we go. Because the sister chromatids are identical to one another, each pole now contains the same DNA and the same chromosome. So sister chromatids are now getting pulled to opposite ends of the cell. These centromeres have split apart. They have been divided, and this chromosome or sister chromatid is identical to this one. Why? Because of DNA replication. We studied that already. And so they're identical. And they're pulled to opposite ends of the cell. And if you're saying, what about mutations? Yes, you're right. Mutations may have occurred, probably did. Usually happens maybe 12 times or something per replication event, and they can be good or bad. Events of mitotic telophase. This is the easiest one. Telo, like telescope, root word for far, far away, meaning the nuclei, the DNA is now far away from each other. Spindle microtubules will disintegrate. Nuclear membrane forms around each group of chromosomes at the poles. You can see that happening here. New nucleus is forming on the top. New nucleus is forming around the DNA at the bottom. Chromosomes will unwind and revert to their extended state. The nucleoli disappeared in prophase, they now reappear. And why? So it's essentially just reversing everything that happened in prophase. We're going to unpack the DNA so that it can be used, kind of like if you're going on vacation. First you packed up your clothes, then you get to your hotel room, then you unpack it so that you can use them. DNA is unpacked so that it can be used. The pinching in of the cell in the middle here is the start of the next phase, which is cytokinesis. The cell will be divided in the middle like that. Cytokinesis in animals. Microfilaments attached to plasma membrane at the equator of the cell. Contraction of this ring pinches off the membrane, forming two cells. This ring here is sometimes also called a contractile ring because it contracts and it's a ring. So it gets smaller and smaller and smaller and smaller and smaller until pop, you get two new daughter cells that are genetically identical to each other. And this is a real picture of cytokinesis occurring. All that fuzzy stuff around it, that's basically proteins, glycoproteins, molecules that are on the outside of the cell membrane. Cytokinesis in plants. I've attached an animation on Blackboard, so you can take a look at that. Basically what happens is, number one, the cell walls are too stiff to be pinched inward, so we can't get that contractile ring mechanism to work with a plant cell. So they just basically build a new cell wall and build a new cell membrane in the middle of the cell. Carbohydrate-filled vesicles from the Golgi will fuse in the middle of the cell, first forming a cell plate and then forming a cell wall. What's a cell plate? It's basically an immature cell wall. It's a whole bunch of carbohydrates that are binding to each other and getting organized, and eventually we get a cell wall forming. So the cell wall forms. You can see it starting to form in the second phase here. That's vesicles being transported there. They start fusing. Even here, so now we have a cell wall in the middle, and then basically just a new cell membrane will be synthesized on either side of that cell wall, like so, connecting the cell membrane on the left to the cell membrane on the right. So now the whole cell is surrounded by a cell membrane. 3 new cell membranes formed on either side of the cell wall or cell plate. Body, heal thyself. So, because stem cells produce identical daughter cells, Bartolo Colon's pitching arm was healed. We think. We're not sure. Scientists are learning to control and stimulate the cell cycle for therapeutic applications, such as lab-grown organs and tissue repair in vivo. In particular, burn victims. There's a link on Blackboard that shows a really interesting video of how we're using stem cells to treat second and third degree burns. It's still experimental last time I checked. Another experiment, an old cow was injected with a culture of its own stem cells. So that means the researchers took a sample of the cow's stem cells out of the cow, put it in petri dishes, stimulated these cells to divide so that there's more of them, and then they injected those cells back into the cow. So that way, there's no possibility of the cow rejecting them as a foreign agent. So what happened? The cow, it regained some characteristics of youthfulness, such as increased mobility. Then cancer showed up all over its body and it died. This is because the stem cell's cell cycle went uncontrolled, which leads to cancer. So stem cells, when they are controlled properly by the body naturally, they divide only a certain amount, just enough to repair whatever tissues have been damaged. But if the cell cycle goes uncontrolled, so for example, we do this crazy experiment and inject stem cells into an old cow, sometimes that can also lead to cancer. So Bartolo Colon's treatment was not without risk at all. Oh, sorry. Now it's in white. I don't know how that happened. Control of the cell cycle. Firstly, let's all take a moment to just appreciate how cute this dog is with his huge floppy ears. All right? Okay, now moving on. Growth factors. They are secreted in response to the need for cell division. So, for example, in response to injury, when the body is growing, or in response to exercise. So you cut your elbow, broken cells, factors inside of those cells that have been broken, some of them will be growth factors, and they'll stimulate nearby stem cells to divide, and not only to divide, but to divide into the correct type of cell, i.e. skin cells. All kinds of other cells will have to be grown, right? Like all the veins and little arteries called arterioles and venules, all those will also have to be produced, all controlled by growth factors. Increased synthesis of cyclins, which regulate enzymes that trigger events such as DNA replication. What was I going to say? Right. The reason that dogs lick their wounds is that their saliva contains growth factors. So the licking of the wound means that that will stimulate the cells nearby the wound, peripheral to the wound, to start dividing and healing the tissue. No, you should not lick your dog's wound. That's not the same thing. And also, no, you should not let your dog lick your wounds. I think that goes without saying, doesn't it? Checkpoints control the cell cycle. So there are 3 major checkpoints in the eukaryotic cell cycle, and each is regulated by protein complexes. So lots of proteins, and they can bind to various things, they can respond in various ways, and essentially they say, are we ready to divide, and is everything right? No mistakes have happened. So for example, there's one checkpoint that it controls the cell from going from G1 to S when some kind of mistake has happened. So basically, is the cell's DNA intact? For example, are all the chromosomes whole? Sometimes they fracture into 2 pieces, and that's a problem. So the cell would have to either repair the DNA, or it would have to, sometimes they'll undergo what's called programmed cell death. If the cell determines the DNA is just broken beyond repair, it will essentially commit cell suicide, is what it's called, or programmed cell death. G2 to mitosis. So if there's no problems going from G1 to S, then the cell goes to S phase, replicates its DNA. Then from G2 to mitosis, has the DNA been completely and accurately replicated? So that means every chromosome has been replicated. There haven't been any major mutations, and each chromosome is attached to its sister chromatid. So there we go. It's attached to its sister chromatid by a centromere. Metaphase, anaphase. Are all the chromosomes attached to the spindle and aligned properly at the cell equator? So are they all lined up like so at the cell equator so that they can all be pulled in opposite directions? Are they all lined up? Have they all had their kinetochores bound to a microtubule, etc.? Are we ready to segregate the chromosomes? And everyone likes cartoon. G2 to M, have you completed your DNA replication? It's kind of like running a stop sign. You can't run the stop sign. You need to stop and make sure you've completed DNA replication. If you have, then you could go to metaphase and then continue to divide. You divide, then you're in G1 doing it whatever it is you're supposed to do. And then if you get stimulated to divide, the cell is going to check. Is all your DNA intact? Because we can't just have cells that have all these mutations replicating, because they will, if their DNA is damaged, then they will not be able to perform their differentiated function correctly. A little more detail now on cancer. Cancer is what happens when we get an uncontrolled proliferation of cells. So for example, when one or more of those checkpoints we just discussed is not functioning, then we get an uncontrolled proliferation of cells. It would be like as if somebody just took all the stop signs away in a city. You're going to get total chaos. It's not good. What causes cancer? Mutations, or specifically mutagens, so those are toxins, UV light, or carcinogens specifically. They cause mutations, which cause uncontrolled proliferation of cells if you actually get cancer. So mutagen and carcinogen, they're basically the same thing in that they all cause mutations, but we identify some of these things that cause mutations as carcinogens if they have a tendency to lead to cancer. So for example, some carcinogens we are all probably kind of aware of: tobacco smoke, lung cancer; UV exposure, skin cancer; radiation, all kinds of cancer, like don't go near a nuclear meltdown, if that happened. And even infections like HPV that can sometimes cause cervical cancer, and also rectal cancer, and in rare cases, penile cancer. Not great. So normally, cells divide. We get an increase in cell number. And one kind of gene called it can be called a proto-oncogene. Proto means before. These oncogenes will stop cell division. So we can see that this cell mass, it's reached, I'm going to tell you, we're just going to assume it's reached its correct size. All right. So maybe it's a finger, and okay, the finger's big enough. One of these oncogenes is going to say, all right, cell division, stop. So we get normal cells, a normal sized finger, for example. But if the oncogene itself has been mutated, so an oncogene is going to be one of the things that participates in those cell cycle checkpoints. If it's been mutated, we can call it an oncogene. So from proto-oncogene to oncogene. And in this case, it just doesn't do its function. It doesn't say stop dividing. Cell division just keeps continuing and continuing and continuing. Another type is a tumor suppressor gene, and they essentially do the same thing. Tumor suppressor genes, we could think of them a little bit differently. This is like a second line of defense, and your book goes into a lot more detail, which I will not be quizzing you on. Just kind of the general role of a tumor suppressor gene. Tumor suppressor genes stop cell division, and thus prevent the formation of a tumor. So normally, this is what happens normally. Cell division will stop when these tumor suppressor genes turn on. But if the tumor suppressor gene has had a mutation or multiple mutations, then it doesn't turn on. And then we get a continuation of cell division, and the production of a tumor. Tumor formation and metastasis. So tumors are rapid, repeated cell cycles, each with a very short interphase, so they also don't grow very much. Tumor cells are typically smaller. They're also undifferentiated. So it's not like if you get a tumor in your finger, you're going to get a giant finger, a really long finger. No, it'll become an undifferentiated mass that will interfere with the normal functioning of nearby tissues. Now, benign tumors, they are unable to spread. For example, maybe if you've ever had a wart on the surface of your hand, very likely a benign tumor. It can't spread. Why is that? Well, because it's difficult for a cell to go from one place in the body to another place in the body unless it's programmed to do that, as some cells are. Malignant tumors, they metastasize. Metastasize means to spread to other parts of the body. There's a link here, but it really has a lot more details than you need to know. So just what's in the book is totally enough. If you feel like checking out this link, you're welcome to. The spreading of cells goes to different parts of the body, and then another tumor forms. It's called a secondary tumor. And these secondary tumors, if they form in, say, the lungs or the heart or the brain, they're going to interfere with what those organs do, interfere with breathing, interfere with what the brain does, and it will eventually lead to death because they just continue to grow. Okay, I already said that. These are epithelial cells of the cervix, and one of them is cancer, one's not. So which cells are, and how do you know? You can pause it and think about it for a moment. Okay, if you didn't pause it, well, the cells on the left are bigger, the nuclei are smaller. The cells on the right are smaller, and the nuclei are bigger. So the ones on the right are from a malignant tumor. And so tumors, remember, are undifferentiated cells. Cancer is undifferentiated cells, and they're usually smaller. They divide rapidly, so that's one of the reasons they're smaller. Their G1 growth phase is very short. They basically just go through a mitotic division. G1 is super short. They go into S phase where they replicate their DNA, and then they divide again. So tumor cells are smaller, they're undifferentiated, and they perform no useful function other than using up nutrients. All right. The next slide is the last slide, and I'd like to just mention that if you are a bit squeamish from looking at real pictures of tumors that have been removed or kind of icky pictures, you might want to just close your eyes for this one. Pardon me. Because we're going to look at two tumors, one removed from a testicle and the other one removed from an ovary, and these tumors are called teratocarcinomas. Terato is Latin for monster, and these things are a little scary looking. So I'm going to go to the next slide now, and if you want, you can just not look at them, and you're definitely going to get all the information you need to get by hearing. So just a little warning, this next slide's a little gross. So here we go, two teratocarcinomas, one from a testicle. The one on the left is from a testicle, and you notice that it has grown a small arm with some very small fingers. The one on the right is from an ovary. It is larger, and it has grown teeth and even hair. When these things are removed, they sometimes will even respond to touch. Sometimes they have something like a heartbeat, and they are very scary looking, and that's why they're called teratocarcinomas. They're very rare. Consider what we have learned about differentiation. Sex cells, so these are both cancers of sex cells, and the sex cells lead to the formation of a zygote in healthy tissue, right? When reproduction occurs, sperm and egg meet, and then that cell is totipotent. It can divide to produce all cell types. So if in cases, very rare cases, where cancer occurs in the reproductive cells, these cancers, these tumors, will actually differentiate into nervous tissue, cardiac muscle, teeth, hair, arms, everything. And so the reason for that is that it is essentially cancer of stem cells that can divide to produce all kinds of cells. All right then. So I guess we didn't really finish this lecture on an upbeat note, but I hope you found this lecture to be interesting, and I'll see you in the next lecture.